present in the scrotum. They develop in the abdo part through intermediate mesoderm of, yes, they are derived from the intermediate mesoderm from the gonadal ridges and from the migrated primordial germ cells from yolk sac. Then pair of testes, genital ducts. What is the genital ducts? Epididymis, vasa deferens, and ejaculatory duct. Associated glands, prostate, seminal vesicles, and corpus or bulbourethral gland. And then we have, see, these germ cells are derived from the primordial germ cells. So first there is differentiation of these cells into the dark type A spermatogonia. Then they differentiate into pale type of A spermatogonia. Now this A spermatogonia turns into the B spermatogonia, which undergoes mitosis on form primary spermatocytes. Primary spermatocytes. Then we have meiosis. Undergo meiosis 1, form secondary spermatocytes. Secondary spermatocytes then form, yes, undergo meiosis 2, and then there are haploid DNA material in each of these 4 spermatids. Now maturation of spermatids into spermatozoa, basically they are undergoing change of shape, which is known as metamorphosis, that takes place, and it is known as spermiogenesis. So these stages are taking place here in from the basal lamina of seminiferous epithelium, which is stratified germinal epithelium. What are the 2 types of cells there? Sertoli cells and spermatogonia. So quickly revise what we have discussed till now, that male reproductive system has pair of testes. It develops in the abdomen, descends down later in life into the scrotum, and then we have genital ducts, associated glands, and penis. Then the structure of testes. So testes is, it is present in a sac derived from the peritoneum, which comes along with it during the descent, which is tunica vaginalis. Then the inner capsule is tunica albuginea. Posteriorly, this tunica albuginea is thickened and it forms mediastinum, testes, this one. The rete testes are present in the mediastinum testis, so it's not mentioned here. So posteriorly we have mediastinum testis, is a thickened part of tunica albuginea, and in the innermost loose connective tissue layer is tunica vasculosa, which has these blood vessels. Now inner part of this testis, it is divided into number of lobules by the presence of septa extending from the mediastinum testis. Now each of these septa have 1 to 3 seminiferous tubules. These seminiferous tubules are lined by seminiferous epithelium, which has 2 types of cells. This we have discussed. And now spermatogonia, these are protected by the lateral process or the junctional complex of the surrounding Sertoli cells, forming blood testis barrier, which protects the spermatogonia from the outer environment and from immune system. So these are the layers that we have discussed. And then mediastinum testes, and uske baad, you know the course of this duct system. These seminiferous tubules then form straight tubules. These straight tubules enter into the mediastinum and forms rete testes. Rete testes then form 12 to 15 efferent ductules, which enters into the epididymis. This comma shaped part here, this is epididymis. It has a head part, body part, and tail part. Then it continues into the ductus deferens, which unite with the duct of seminal vesicle to form ejaculatory duct, which opens into the pineal urethra. So seminiferous epithelium. Now please be quiet, we are starting with the histology part. So these are the cut section showing a different seminiferous tubules. These seminiferous tubule has 2 types of cells, again germinal cells and Sertoli cells. Now how to identify these cells? Sertoli cells are long columnar and they have this ovoid nucleus towards the basal region, and spermatogonia are in different stages of spermatogenesis. In single section, all these stages may or may not be visible. So the length of seminiferous tubule is around 50 to 80 centimeter, highly convoluted, and it is lined by specialized germinal epithelium. This is cut section of seminiferous tubule. Now let us talk about Sertoli cells. These are tall columnar cells. Now can you identify which one are Sertoli cells? The one which are painted yellow here. These lightish brown beige color. They have a ovoid nucleus and they form junctional complex and divide this compartment into two parts. The lower compartment where spermatogonia and primary spermatocytes are there, this compartment is the basal compartment. The upper compartment which have further stages of spermatogenesis is L-lumen compartment, luminal compartment. Okay, so these are projecting towards the luminal surface. Now this is blood testis barrier that we were talking about. These are two columnar Sertoli cells. This lower basal compartment having spermatogonia, and these are the later stages. So question arises how these crosses this blood testis barrier. These are present below basal compartment and other cells are present above it. So how they diffuse into this? So during spermatogenesis, this is loosened up. This junctional complex is again restructured after once the spermatogonia further pass into the lumen compartment. So there is remodeling of this junctional complex after formation of spermatocytes. There are apical cytoplasmic processes and lateral cytoplasmic processes. This junctional complex is between the lateral cytoplasmic processes of Sertoli cells. So what are the functions of Sertoli cells? Yes, we always say nutrition and protection. Any other function that you FSH secretion नहीं करता है, FSH receptors होते हैं इसके ऊपर, तो जब भी bind करता है FSH, तो then there is secretion of inhibin, which inhibits the secretion of FSH from the anterior pituitary. Yes, so secretion of inhibin first. Second, any other factor? It secretes anti-Mullerian hormone or substance, which what does it do? Regresses Mullerian duct and stops the development of female reproductive part. Then any other factor that it releases transferrin, which helps in the transport of iron, and androgen binding protein, which helps in concentration of testosterone. And obviously supporting nourishing functions are there, and also it phagocytoses the residual bodies that remains during the spermiogenesis of spermatids into the spermatozoa. So they are supporting cells, they are nourishing the spermatogonias, the germ cells, establishing the blood testis barrier, phagocytosis of residual cytoplasm, and secretion of androgen binding protein, anti-Mullerian hormone, inhibin, transferrin, and it also provides fructose rich medium. That means it is giving nutrition to the germ cells. So these are the functions of Sertoli cells. Now what is the function of this blood testis barrier? Why we have required? Those who have came late, Sertoli cells form this blood testis barrier. So this basal compartment which has the interstitial space, which has spermatogonias, and above compartment is luminal compartment. Now this is separated from the outer environment and it protects the developing cells from the immune system. There may be these are regarded as these are haploid spermatids, and they may be attacked by your own immune system in the form of attacked by the antibodies against these haploid germ cells. Okay, then cut section of seminiferous tubule showing different stages of spermatogonia. तो सबसे luminal surface, सबसे अंदर की तरफ क्या present होंगे? Spermatids. And middle part में जो largest cells होते हैं during this cycles are spermatogonia B. These are present in the middle part of this stratified layer, and supporting cells are there, tall columnar Sertoli cells. So this is the cut section of individual seminiferous tubules showing the stratified layer and different types of cells. Now these cells are surrounded by a thin muscle cell layer, smooth muscle cells layer, which are myoid cells. Okay, these myoid cells helps in the contractions and expulsion of sperms as and when required. इतना समझ में आया सबको? So अगर आप हिस्टोलॉजी देखोगे इसकी तो क्या देखोगे इसके अंदर? अच्छा, वी हैव ऑल्सो वन दिस, इतना क्लास डिस्टर्ब हुआ है आज, So in between these connective tissue, we have important polygonal cells which are there, which are known as Leydig cells. What is the function of this Leydig cells? Yes, the testes have germ cells and also production of the spermatozoa and secretion of testosterone. So testosterones are produced by Leydig cells. And what is the location of Leydig cells? It is present in the loose connective tissue surrounding the seminiferous tubules. What is the shape? Polygonal shape. And how you gonna recognize this? They have a special this eosinophilic stain because they have abundant amount of smooth endoplasmic reticulum. So they are stained pink in color. So these cells are present here in the interstitial part in between the seminiferous tubules. So this is the cut section of the testes showing number of CS sections of seminiferous tubule in stratified manner surrounded by the myoid cells, smooth muscle cells, and in between there is connective tissue layer derived from tunica vasculosa. Yes. So this is the hand-drawn diagram of testes showing columnar cells and spermatogonia in different stages and surrounded by the loose connective tissue. So what we have discussed in the histology of testes, let's just concentrate on the histology. There are three layers: outermost serous sac, which is tunica vaginalis. Then we have dense connective tissue layer, which is tunica albuginea, and posteriorly it forms a thickened part, which is mediastinum testis. Then inner part we have a thin connective tissue layer, which is tunica vasculosa. Talking about the epithelium, it has basal lamina has two type of cells: large columnar Sertoli cells, and we have discussed their functions. So you must remember their functions. What are the functions of Sertoli cells? Once you recognize them, you must be able to elaborate their function. So supporting, nourishing to हर कोई you will say it. Supporting cells, blood testis barrier, yes, and inhibin secretion. Inhibin क्या करेगा? It will stop inhibits FSH secretion from anterior pituitary. Anti-Mullerian hormone, which will regresses the development of Mullerian duct. Yes. Transferrin, iron transporting protein, and androgen binding, which concentrates testosterone, and phagocytosis of the residual cytoplasm. This is the bookish diagram, this is the hand-drawn diagram, and this is the microscopic picture. Now the duct system, epididymis and ductus deferens. So epididymis starts at the posterolateral surface of testes. Again, let's discuss those ducts. Straight tubules, seminiferous tubules bleeds into straight tubules. Straight tubules, they have low columnar to cuboidal epithelium. Then we have arete testes, again cuboidal epithelium. And then we have efferent ductules. 12 to 15 efferent ductules opens into the head of epididymis. This epididymis has a upper head part, middle body part, and the lower tail part. It is comma shaped. And main function is, once these sperms are discharged here, they are stored in the epididymis and ejaculated as and when required. So here, what are the things that is taking place? First of all, it is a reservoir of sperm, yes, and partial motility to the sperms, and it also provides a glycoprotein coat over the sperms, which is decapacitation factor. So it protects the sperm. Only the complete motility of sperm is acquired once it is lodged into the female reproductive tract. Yes. So epididymis, again, when we will see the cut section, because it is again a convoluted duct system, it has different lobules, and these lobules have pseudo-stratified ciliated columnar epithelium. अभी तक आप कहाँ पढ़ रहे थे pseudo-stratified ciliated columnar epithelium? Respiratory system. So vasa deferens and epididymis are other best example for pseudo-stratified ciliated columnar epithelium. So here in the cut section of epididymis, you are going to see numerous tubules which have these pseudo-stratified ciliated columnar epithelium. So it is having two types of cells: tall columnar principal cells and small basal cells. As you have read, these basal cells, basically they are stem cells. Now they are stem cells and they are differentiating into the primary cells, that is principal cells. So these principal cells have cilia. These are modified non-motile stereocilia. So stereocilia is exclusive feature of male reproductive part of vas deferens and epididymis. क्या present होगा यहाँ पे? On the upper part of the on the surface of principal cells, they have stereocilia. Now, what is the function of stereocilia? The sperm is transferred from the testes into the epididymis. It comes along with the fluid secreted with in the testes. So it absorbs excess of fluid. Stereocilia, what does it do? It absorbs the excess fluids which comes along with the sperm, and it generates a current system through which sperms are passed on to the tubules. Yes, they are non-motile. They are not helping in the mobility. They are non-motile, modified microvilli only, and what our major function is absorption. And they are providing partial motility. How? They are basically generating a fluid current mechanism. They are absorbing a lot of fluid. It's coming with a lot of pressure. It's also pushing forward the sperm. So basically, stereocilia are, what are they? These are modified non-motile microvilli present on the surface of principal cells. So what is the epithelium of epididymis? So it is around 4 to 6 meter in length. It has a head, body and tail. This is the cut section of epididymis. You are going to see a lot of these tubules. They are cut. See, you are going to see same picture as in testes. If you are not able to differentiate the epithelium, आपको testes में भी ऐसे lobules दिख रहे हैं, इसमें भी ऐसे lobules दिख रहे हैं, तो आप किस पे फोकस करोगे? फिर एपिडाइडमिस में आप सरफेस सेल्स को देखोगे, यहाँ पे प्रोजेक्शंस हैं बहुत सारे यहाँ पे. These are stereocilia which are not there. And again, what is the function of major function is storage of sperms. So in the lumen you are going to find collection of sperm in the luminal cavity of epididymis. So this is the identifying feature for epididymis. Yes, and then again these are lined by outer layer is of smooth cells. Smooth cells, what are they doing? Smooth muscle cells. They are contracting and providing mobility and pushing the sperms forward into the, what will be the next? Vas deferens. So vas deferens is again the passage through which sperms pass into the ejaculatory duct. And now ejaculatory duct is formed by the combination of vas deferens and the duct of the seminal vesicle. Now again, the vas deferens has a pseudo-stratified ciliated columnar epithelium. This is the microscopic view of epididymis. Pseudo-stratified ciliated columnar epithelium, two types of cell, basal cells and principal cells. Principal cells have stereocilia and they are surrounded by a thin layer of smooth muscle cells which helps in the, they contract and push the sperm forward. So basically helping in the mobility of the sperm. And inner luminal cavity, it has collection of sperm. In some books, there are a third type variety of cells is also there. They are known as halo cells. These are basically the lymphocytes which are present in the epithelium of these epididymis. Halo cells are type of the lymphocytes which are present in the epithelium. So there are three types of cells: basal cells, principal cells, and halo cells. Next, we have this is hand-on diagram. जो आप सूडो स्ट्रैटिफाइड एपिथेलियम बनाते हो, यस, कहाँ बनाया था अभी आपने? रंगोली में, ट्रैकिया में. सूडो स्ट्रैटिफाइड का मेन फीचर क्या है? इसके अंदर कैसे आइडेंटिफाई करोगे सूडो स्ट्रैटिफाइड? व्हाट इज सूडो स्ट्रैटिफाइड? व्हाई दे अपीयर डिफरेंट लेयर्स? लेंथ डिफरेंट होती है, पर क्यों? लेंथ तो डिफरेंट है, उससे क्या मतलब? न्यूक्लियस का लेवल ऊपर नीचे है, क्योंकि लॉन्ग सेल्स होंगे उनका न्यूक्लियस लेवल ऊपर होगा, शॉर्ट सेल्स का न्यूक्लियस जो होगा वो नीचे होगा, इस वजह से वो स्ट्रैटिफाइड अपीयर होता है. ठीक है. नेक्स्ट वी हैव व्हाट वी हैव नेक्स्ट, vas deferens. Now vas deferens slide is totally different from these epididymis and testes. It has this mucosa, lamina propria, and three layers of muscle cells, muscle layers. These muscle layers are arranged into inner longitudinal, middle circular, outer longitudinal. Now there are three muscular coat here in the vasa deferens. Why? The answer is you already know. When they contract, they will provide peristaltic contraction or the movement, which will help in the movement of the sperm. So that is why there are three layers of smooth muscle cells here in the vasa deferens. Now the here key feature of vasa deferens is the star-shaped lumen. Where you have studied star-shaped lumen? In the ureter, very good. And also there are foldings in the esophagus. I told you there are mucosal folds, but don't memorize it as star-shaped lumen, because these two have uh, their principal feature is star-shaped lumen. One is ureter and second is vasa deferens. Ureter epithelium in the ureter is transitional epithelium, and here epithelium is pseudostratified ciliated columnar epithelium. These cilia are basically stereocilia. Function I have already explained. So what you are going to see here, there are longitudinal folds which is giving star-shaped appearance to the lumen. Yes, so there is a star-shaped lumen lined by pseudostratified ciliated columnar epithelium. Again, there are two types of cell, long principal cell and short basal cell, and then there is outer loose connective tissue layer which has these blood vessels and lymphatics, which is lamina propria, and then outer layer is muscular layer in the form of three layers: inner longitudinal, middle circular, and outer longitudinal. Contraction of these muscles provide peristaltic movement to the peristaltic movement for the movement of the passage of the sperm. And the outer adventitia layer is there. Outer adventitia layer is basically loose connective tissue layer having blood vessels and lymphatics. So this is the histology of vas deferens. Star-shaped lumen, it will appear as if it is slide of ureter, but no. How you are going to identify then? Going to see epithelium, transitional has stratified layer, and here there is pseudostratified ciliated columnar epithelium, and outer, this muscular coat is really thick. It is in the form of three layers: inner longitudinal, middle circular, and outer longitudinal. This is hand-drawn diagram. Star-shaped lumen, pseudostratified epithelium, lamina propria. Then we have three layers of muscles, smooth muscle cells, and outer adventitia layer. अभी तक हम वो लोब्यूल्स देख रहे थे, लोब्यूल्स में एपिथेलियम देखे थे, और अभी हमारा वहाँ पे सब क्या आ गया? म्यूकोजा, लैमिना प्रोप्रिया, मस्कुलर लेयर एंड एडवेंटिशिया. Then accessory gland, prostate is the largest accessory gland. It is pierced by ejaculatory duct. Basically, this gland surrounds the penile urethra, and this ejaculatory duct opens into the penile urethra. So it is pierced by the ejaculatory duct. What? Prostate gland. It has two types of capsule: outermost false capsule, which is derived from the pelvic fascia, and the inner true capsule. Then this true capsule is having smooth muscle cells and fibers layer, collagen and elastic fibers, which are also embedded into the stroma of prostate gland. So basically, the parenchymal cells these are surrounded by fibromuscular stroma. Fibromuscular stroma मतलब क्या हुआ? Fibers are there, collagen, elastic fibers, muscular, smooth muscle cells are there in the surrounding medium of parenchymal cells. Now here the cells are basically alveoli, which are forming tubuloalveolar gland. There are 30 to 50 tubuloalveolar gland in the prostate. Now here these glands are divided into three parts which are surrounding the penile urethra. The innermost, which is surrounding the penile urethra, is the mucosal gland, which opens directly into the pineal gland. Then the intermediate part is having submucosal glands, which opens through the duct into the pineal urethra, and the largest one, the main prostate gland, are the outermost, that opens again through the duct into the penile urethra. In the cut section, the prostate will appear like this. There may be numerous alveoli. The epithelium of alveoli, it varies from pseudostratified columnar epithelium to columnar epithelium. There may be simple columnar epithelium depending upon the secretory activity of the gland. So what is the contribution of this gland? Yes, 20 to 30% contribute to the semen. Yes, and the main content is fibrinolysin, which helps in the liquefaction of the semen. They also have citric acid and specific antigens and acid phosphatase. So these are basically the substances that are secreted into the semen by the prostate gland. The main content here is fibrinolysin. What is the function of fibrinolysin? Liquefaction of semen. And where this secretion is discharged into the penile urethra, where the ejaculatory duct opens. Then the next we have the outer peripheral fibromuscular stroma, having connective tissue layer consisting of collagen fibers, elastic fibers and smooth muscle. And these smooth muscle cells, again, they are helping in the contraction of these tubuloalveolar gland for the secretion of the fluid into the penile urethra. Now here one important part is there. Can you recognize these concrete part here, the pink colored one, into the lumen of these glands? See, these are the tubuloalveolar gland, which have these pyramidal shaped cells having the oval nuclei, and they have these rounded concretions here. As the age increases, advances after 50 years, you may find these collections into the lumen. ये हमने कहीं और भी डिस्कस किया है, where we have discussed there are these concrete laminated structure which are present in the, yes, anyone? No. उसका हिस्टोलॉजी आपको दिखाया नहीं है, लेकिन क्लास में मैंने बार बार बताया था. It starts with corpora, erinacea, brain sand. Brain sand is present in third eye, intuitive power, pineal gland, biological clock. So corpora erinacea there in the pineal gland, corpora amylacea, these are the laminated concrete structures which are present in the old age persons. Yes, and they are basically glycoprotein precipitated and then calcified, and their number increases as the age advances. This is also important feature of this prostate gland. These are present in the lumen of these tubuloalveolar gland. This is hand-drawn diagram for your help. What will be the epithelium? Varies from pseudocertified ciliated or simple columnar epithelium. These are the epithelium of the alveolar gland. Yes, secretion of alveolar gland contributes into the semen, 20 to 30%. Then they are surrounded by fibromuscular stroma. So basically, what are the key features here? Tubuloalveolar gland and fibromuscular stroma. Key content of fibromuscular stroma is smooth muscle cells, collagen and elastic fibers. Blood vessels, obviously, they run here in the connective tissue. An important content of the secretion here, fibrinolysin. Yes. Pineal urethra, what will be the epithelium of pineal urethra? These structures surrounding the pineal urethra, and ejaculation is done into the pineal urethra. The upper part of pineal urethra, which is continuing downward from the urinary system, is lined by transitional epithelium. The lower part is stratified columnar epithelium. Okay? So upper part of pineal urethra is transitional epithelium, and lower part of pineal urethra is stratified columnar epithelium. In both the section, there is no histology here shown of the pineal urethra. You may or may not see in the section of this prostate gland, but I think we have a slide there which has this pineal urethra piercing the gland. Okay, their epithelium may be transitional or what? Stratified columnar, depending upon the section, which section has been taken. Next, we have seminal vesicle, another type of gland, mostly contributed part by seminal vesicle into the semen, 70%. Important content of the fluid contributed by it is fructose-rich medium, which provide the energy to the sperm, and also phospholipids. Then, ये देख के can you explain now histology? Anyone? अगेन नाउ आपने इतनी हिस्टोलॉजी तो पढ़ ली है कि आपको ये पता है कि इनर लेयर क्या है, आउटर लेयर और उससे आउटर मोस्ट लेयर क्या है. तो नाउ कैन यू एक्सप्लेन मी इनर लेयर? यू आर सेइंग दीज, yes, irregular mucosal surface. So this mucosal surface is thrown into numerous folds which are anastomosing with each other, forming crypts and cavities. So it has the most like we have discussed all the slides. सबसे ज्यादा इरेगुलर जो ल्यूमेन अपेयर होता है वो इसका होता है सेमिनल वेस्ट, क्योंकि ये हाईली कॉन्वोल्यूटेड gland है. So it has this mucosal folds which anastomose with each other, branching is there, cavity formation is there. So it appears like this. Yes, then what will the epithelium? Yes, it varies from pseudocertified to simple columnar epithelium. Then we have lamina propria and then the layer of smooth muscles and outer adventitia. Last we have organ of copulation, histology of penis. Now there are 3 erectile tissue, 2 present on the dorsal section and 1 present in the ventral section. The dorsal section one are known as corpora cavernosa, and the interior one or the ventral one is corpora spongiosum. So these larger caverns are filled with there is blood here which is supplied by the helicine artery. Yes, it happens when there is neuropsychological stimulation. There is excess blood supply to the gland which leads to expansion of the cavernous spaces and which compresses the venous system present behind it. So how does it happen? These cavernous spaces, these are surrounded by tunica albuginea. This tunica albuginea is thicker as compared to the tunica albuginea surrounding the corpus spongiosum. This is sponge-like a part here, erectile tissue present interiorly. It is pierced by, okay, this is that we were discussing there in the prostate gland, that was the prostatic urethra. My apology, because the prostate gland is surrounding the prostatic urethra. Here spongiosum is pierced by the penile urethra. My apology, I am really sorry. हो जाता है, ठीक है. So prostatic urethra, it has 2 type of epithelium. The upper part is transitional and the lower part is stratified columnar. And here again we have, it has again 2 types of layers, simple or stratified columnar epithelium, and the lower part which is near to the skin, it is stratified squamous epithelium. Now the blood vessels which are present here within the cavernous space, this is the deep artery, and there is a branch of this deep artery which supplies this cavernous space, helicine artery. So as when there is stimulation, this artery gives more blood supply and the expansion of these cavernous spaces is there, which leads to compression of the venous blood. So there is more excess blood present here and erection of penis. Then anteriorly we have corpus spongiosum. Again it is surrounded by tunica albuginea, and the inner part, it has a wavy part which is basically opening of the penile urethra. Penile urethra has 2 epithelium. The upper layer is stratified to simple columnar epithelium and the lower one is stratified squamous epithelium. Now the outer capsule of this penis is basically buccal fascia. This is the connective tissue layer which is devoid of fats. So fascia of this penis is known as buccal fascia. And then outermost layer will be skin, which is stratified squamous keratinized epithelium. Thank you. So let us revise what we have discussed quickly. Only histology part, nothing else. फंक्शंस वगैरह आप लोग जो है मैंने लिखवा दिए हैं, आप लोग वापस रिवाइज कर सकते हैं. ओनली हिस्टोलॉजी पार्ट, हाउ टू रिकॉग्नाइज दीज स्लाइड्स. This is the slide of, this is the slide of again seminiferous. Why seminiferous tubule? Quickly reply please. सबसे पहले स्लाइड किसका दिखाया था मैंने आपको? What is the time? This is the cut section. क्या हो गया भाई? क्या प्रॉब्लम है? हैं? क्या डिस्कस कर रहे हो? सामने देखो, बताओ ये किसका कट सेक्शन है? सेमनिफेरस ट्यूब्यूल. सेमनिफेरस ट्यूब्यूल कैसे आइडेंटिफाई करोगे आप? सर्कुलर लोब्यूल्स तो सबको दिखे हैं यहाँ पर. फिर उस बाद में देखो लोब्यूल क्या होता है. एक लोब्यूल जो है आउटर कनेक्टिव टिश्यू लेयर जो है दैट इसको सराउंड जो कर रहा है. आप लोब्यूल्स में और सेमनिफेरस ट्यूब्यूल में कंफ्यूज हो रहे हो. लोब्यूल के अंदर 1 2 3 सेमनिफेरस ट्यूब्यूल होगी और ये एक कट सेक्शन जो है वो एक सेमनिफेरस ट्यूब्यूल का है, लोब्यूल का नहीं है. लोब्यूल कैसे आइडेंटिफाई करोगे? इसके आउटर मोस्ट वो लाइट पिंक कलर में जो कनेक्टिव टिश्यू दिख रहा है दैट इज कनेक्टिव टिश्यू सेप्टा. दिस वन, this is interstitial connective tissue. So this is basically a one lobule which is having 1 2 3 sections of seminiferous tubule. Now this is one seminiferous tubule. What are the two types of cells here that you are going to recognize? Sertoli cells and yes, germ cells. Sertoli cells are columnar in shape, ovoid nucleus, and they form blood testis barrier. And there is, why it is stratified layer? Because we have germ cells in different stages of spermatogenesis. The lower one are spermatogonia. They differentiate into primary spermatocytes. In the middle region and above region we have secondary spermatocytes and spermatids. And here in the connective tissue part, these cells, eosinophilic staining cells are keep quiet. These are Leydig cells. Function of Leydig cells is androgen secretion. Excuse me, testosterone secretion. Androgen binding protein is secreted by Sertoli cells. Inhibin is secreted by blood testis barrier is formed by anything else. Transferrin is secreted by most important function. Anti-Mullerian hormone is secreted by epididymis. It is again convoluted structures, so you will be going to see these circular structures. These structures are surrounded by myoid cells which are smooth muscle cells. The layer here epithelium is pseudostratified ciliated columnar epithelium. What are they having? Principal cells, stereocilia. This is important feature of epididymis and vasa difference. In the lumen there is collection of swarm. So this basically differentiates it from, yes, testes. Next we have histology of vasa difference. What is the key feature here in the vasa difference? Star shaped lumen. So we have star shaped lumen thrown into the mucosal folds. These again lined by the pseudostratified ciliated columnar epithelium having stereocilia principal cells, and they have basal cells, and then they are surrounded by lamina propria. Important, again second important feature is three muscular layers: inner longitudinal, middle circular, and outer longitudinal. What is the function here? Why these muscular layer are required? Peristaltic contraction. Outer adventitia. Then there are three zones here in the these are surrounding the, this is prostatic urethra, and there are three zones here which are surrounding this prostatic urethra. The inner one is mucosal, then we have intermediate one submucosal, and the outermost which is different from the gross anatomy section. Histologically, we have divided it into three parts: mucosal, submucosal, and main. And the secretion here contributes to the, yes, semen. So basically, main content fibrinolysin function. कितने होंगे? There are 30 to 50 tubuloalveolar glands. These are lined by pseudostratified ciliated columnar epithelium or simple columnar ciliated epithelium. And in the surrounding medium, what you are going to see, there is fibromuscular stroma, which is filled with the collagen fibers, elastic fibers, and the smooth muscle cells, which are required for the contraction of this alveolar gland to secrete the discharge the fluid into the penile urethra, prostatic urethra. Then concrete laminated structures formed after calcification in the advanced stage is known as, yes, these are this pinkish color which are present in the lumen here. These are concretions, prostatic concretion, which are known as corpora amylaceae. Corpora arenacea, brain sand in the pineal gland. This one, this is corpora amylaceae. Highly branched mucosal layer, yes, irregular layer you are going to see in the seminal vesicle, then lamina propria and smooth muscle layer is there, and adventitia. Secretions here contribute into the semen. Then we have penis and penile urethra. There are three erectile tissue, two corpora which are present dorsally. This is the dorsal part and this is the interior part. Then this is corpus spongiosum, these are corpora cavernosa. Okay? There is central artery here in the corpora cavernosa, and here there is penile urethra. And dorsally we have this dorsal vein, dorsal artery, and dorsal nerve. This thickened layer of connective tissue is tunica albuginea, which is partially dividing these two cavernosa. And we have a thin layer of tunica albuginea here surrounding the corpus. And then we have this connective tissue layer surrounding this tunica albuginea, which is known as Bach's fascia. It is devoid of fat cells, and the outermost layer is